Okay, welcome everyone to NSTA Engage Spring 21. And it looks like Chris has started our recording. My name is Heather Ryan and I am this session's moderator. Chris Herman and Rachel Mosey are supporting us behind the scenes today, and Chris Chow will be our presenter for exploring the wonderful world of cell membranes, protein, lipids, and ions, oh my. During the session, please use the chat to communicate. And the chat will then become part of the recording that NSTA posts on the Spring Engage site tomorrow. During the session, Rachel will post links to resources, registration for your student modeling pack, and the session evaluation in chat. I'll also try to monitor chat for your questions, and we will let you know if we have time and you can unmute and interact. I now have the privilege of introducing Chris Chow. Chris holds a bachelor's in bioengineering and a master's in curriculum and instruction from the University of Illinois at Urbana-Champaign. She has been a science educator for 20 years and enjoys facilitating workshops for science teachers in her district, at national conferences, and now virtual sessions. She received the 2014 Spotlight Award for Excellence in Teaching from the St. Vrain Valley Education Foundation and the 2016 Outstanding Biology Teacher Award in Colorado from NABT. She currently teaches AP Biology, Biotechnology, and coordinates the Medical and Bioscience Academy program at Longmont High School in Colorado. And now let's begin the program. Take it away, Chris.
Thank you, Heather, for your warm welcome. And I want to say hi to everybody out there. Looks like from the chat that we have folks from all different time zones. So welcome today. It's still morning here in Colorado. Today, we're going to be spending the next 40 minutes together to learn about the structure and functions of the cell membrane, to explore the role of transport proteins and moving ions and molecules across the membrane. And I'm going to throw in there a case study or story of how the CFTR gene can cause cystic fibrosis. So for those of you who have modeling packs, let's go ahead and get those out and things set up. And let's see, let's get my overhead camera turned on so I can show you some things here. With my student modeling kit, I have went ahead and for my classes, I taught virtually for pretty much most of this fall. And so for my camera settings for showing what these different proteins are, I took my little labeler and labeled what all these different proteins are. So this is available. You'll need to punch these out and then glue or tape them to your cardboard cutout here for the student pack. And then some other things that you're going to see in your student modeling pack is water molecules and phospholipids, and these blue circles and purple squares. And what I did was I went ahead and labeled these. In the classroom version of the water molecules and the phospholipids, you get some red stickers and you can put them on there. But for the student version, I just told students grab a red pen or marker and draw in, these red circles to represent oxygens there or the phosphate group in the phospholipids. And then for the sodium and potassium little dots, it gets really confusing to know what is what. And so blue is the sodium, and so I labeled that, and purple is the potassium. And we'll be using that later in the workshop, so it'll be good to know which is which. The other things in your student modeling pack that you'll need to do is cut out ADP and phosphate groups from the cardboard cutouts and glucose molecules as well. And so in the version that's the classroom pack, you'll see all these in foam, so you won't need to do any punching out or work there. Now, the last thing you're going to see in my workshop that might be kind of confusing is I've handmade some little pink triangles and written chloride ion on it. And as we model the CFTR protein, I'm going to be showing you how the chloride ions are transported across the membrane and what happens if there's a mutation in that particular protein. So those are some things to get you oriented and started with our workshop. Okay, so first thing we want to do is go ahead and take a look at this diagram here of some different representation of phospholipids. What do you notice and what do you wonder? So if you wouldn't mind, participants, if you could type that into the chat, what are some things that you notice about these particular representations, diagrams of phospholipids? Okay, Nicole says model 1 is 3D, nice, okay. The two on the left appear to be the same molecule. Images without words or labels from Iris. Okay, good. Okay. The tails, there are tails that are there. Okay, good. So you notice there are tails. They're molecular structures. Okay, so, and some of you might have noticed that third representation looks a lot like those phospholipids from your kit, the little foam ones that are there. And so you could lay them out and take a look at those. And so that third one is the representation just in our foam models to represent the 3D structure in that first picture or that 2D layout, and both showing you some kind of part at the top, and which you see a lot of that red, which stands for oxygen there, and those long tails, those carbon and hydrogen tails. So let's go ahead and take our foam phospholipid representations from your model. And in the next slide, what I want you to do is take a look at this and also this picture here. So this is a representation of this molecular structure of phospholipids. And to me, it kind of looks like a reindeer with, like, you know, a little short head and long legs. And that head part is the phosphate head, which is polar. So it is hydrophilic and has some negative charges on there and is attracted to water. Those long tails that you see that look like legs, those are made of carbons and hydrogens, which repel water. So they are nonpolar or hydrophobic and have, so this is an interesting molecule because it is amphipathic with both water loving and water hating properties. And so in a watery environment, how do you think that these phospholipids can be arranged? So this is a question that you can ask your students, have them have their phospholipids in front of them, and see if they can think about what are ways, if you understand that there's these parts of the molecule that are attracted to water that are hydrophilic and parts that are hydrophobic that repel water, how might these phospholipids be arranged in a watery environment? So in your modeling packs, there are these cardboard giant beakers, and you could have students, you know, dump these phospholipids into a beaker to show how do you think they might be arranged chemically so that we have what I call matchy-matchy, water loving, attracted to water, water hating, away from water. Okay. And so at home, you may have generated some different orientations of your phospholipids. And so hopefully students can sort of figure out, as we take a look at phospholipid arrangements, there can be several ways in which these phospholipids can be arranged. And I'm going to go ahead and kind of switch to another representation. In the classroom kit, this is showing you one representation of the way phospholipids can be arranged. And so you notice these that are not colored are our phosphate heads that are polar, okay, with the tails facing each other. You could have this kind of arrangement, this lipid bilayer like this. And so, Chris, if you could switch to the next slide there to show the 3 different arrangements of the phospholipids. You can have small circular spheres. Those are called liposomes, and they can be made of 1 or more layers of phospholipids. You could have micelles. Those are the little tiny cluster of aggregate of molecules of phospholipids. And they're really surfactant molecules that are dispersed in liquid solutions. So it's kind of like a colloidal suspension. And then of course, students who've probably seen in their textbooks representations of the phospholipids in 2 layers in that linear way. But I think it's helpful to have the, the big classroom model of these phospholipids to be able to show, as you see on my camera there, to show that these aren't just linear, but they can go all the way around. It's actually spherical in a 3D shape. So that's our lipid bilayer. So as we flip to the next slide, you can see that this is how students often see that representation of phospholipids in their textbook, that cut out from the side. And on the right side of this picture is actually printed 3D models of these phospholipids, which is kind of nice. So you can see it in the molecule with the 3D shape or with the phospholipid representations there. So those are kind of nice to see. But let's go ahead and take a look at a short video of phospholipids in action, in animation here. Okay, so with this animation, you can see that I think of phospholipids, I thought of them before as these really solid structures, kind of like a sandwich. It's kind of hefty. But these phospholipid molecules are really like buoys floating on the surface of an ocean or something like that. And in fact, you could think of that phospholipid bilayer, the cell membrane layer, as like an ocean. And these phospholipids can move side to side. They're sometimes closer, sometimes farther apart from each other. They can also flip occasionally upside down. Now that's not really common, but because their natural orientation is those phospholipid tails facing each other and the heads facing outward to the watery exterior or the watery interior of the cell. Okay. So as we take a look at these phospholipid bilayers or the cell membranes have other things, other components like proteins, some that are embedded all the way through and some that are on one side of the or other. So those are integral versus peripheral proteins. You also have carbohydrates, different kinds of lipids, like cholesterol is also embedded in the cell membrane. So cell membrane is also made up of a variety of different molecules involved there. And so we're going to focus mostly on these proteins that are embedded that go across the cell membrane. So let's go back to our foam model here. Okay, so I've got the student modeling pack in the camera. And what you want to do is go ahead and place your proteins on there, and we can see there's a variety of ones that we can demo through just the student pack. The classroom version allows us to be able to have movable foam pieces and larger, this particular yellow representation of phospholipid bilayer here. That's in the classroom pack, and you can make ginormous cells on surfaces of larger science tables there. But this is pretty handy, and you could see different channels that are involved in here, and we're going to kind of briefly go through all these today in today's session. So let's go ahead and to the next slide there with the membrane transport proteins. And so with these variety of proteins that are embedded in the cell membrane, and they can allow substances to cross. So we can have potassium ions and sodium potassium pump, transporting glucose across the membrane, water molecules through aquaporins, and sodium channels as well. So these different proteins can either be channel proteins or carrier. Channel proteins include this list of examples. They allow an open pore basically to allow these substances to cross, versus carrier proteins. They loosely bind to the molecules they are transporting across. Some require energy, like an active transport. So anything with a pump name requires energy in the form of ATP. And so as we take a look at the graphic on the next slide, you can see how channel proteins just kind of provide an open channel or tunnel for things to cross, and carrier proteins actually change the conformational shape so that it loosely binds those molecules to move them across the cell membrane. So let's start with these, some of these proteins that move without the use of energy, and that's passive transport. And so this graphic shows how all of these molecules are moving from an area of high concentration to low concentration. So they're traveling down their gradient. Really small molecules like gases can simply diffuse across, don't need that channel protein. And then other molecules that are ions or some larger things like glucose will need the help to get across because of their polar properties. It's difficult to cross that hydrophobic lipid bilayer with those hydrophobic tails there. So let's talk about one of the biggies, which is aquaporins here. And in my overhead cam, I'm going to go ahead and show my 3D model of aquaporin right here. And one thing you'll notice is that aquaporins, they have these alpha helices. Oops. They have these alpha helices. You can see these curly Qs kind of going down. And these have R groups or side chains that are hydrophobic. So it's nice to be able to match with the hydrophobic tails and anchor into the cell membrane. And what's fun about this 3D molecular designs model is that there's a portion of this that can be removed. And so you could see that removing part of the structure to see the interior of this channel, which allows these little water molecules to cross. So this is one of my favorite 3D models. I like to pass that around, and then the kids pass it around, and then very carefully, of course, they realize it's magnetic, and it's really fun to open that up and see the interior of the protein there. So aquaporins are for transporting water across because water's polar, clearly, and difficult for them to cross. Some water molecules can cross, but it's at such a low rate, cells simply could not survive with just the diffusion, simple diffusion of water. So there's aquaporins to help transport water across. There's actually 13 different known types of aquaporins, and they're found in all kinds of cells. In the body, including kidney, lung, blood cells, skeletal muscle, and then plants and microbes also have aquaporins as well. So the next slide shows that I showed you a 3D model of one unit of aquaporin protein, but aquaporins actually form a tetramer or 4 subunits together. And you can see that cluster of them in that top view on the right side, the David Goodsell graphic there shows that there are 4 aquaporin molecules or subunits, and each one of them is a channel for water. Okay, so let's go ahead and take a look at this video, which is really neat to show the movement of water molecules in an animation. All right, and you can see this channel provides a like 1-by-1 single file line width of channel for water molecules to go through. And these R groups or side chains help to facilitate the movement of these molecules. And water can go in both directions. So it's not just a one-way flow. Water can flow in both directions from where there is a high water concentration or water potential to low water potential. So if we were to let's go ahead and take a look at our model of aquaporins. I'm going to switch to the classroom version right here, if you're going to see that on my overhead cam right here. Okay, and this channel right here represents the aquaporin. Actually, I'll flip it that way. And then what you could do with your foam pieces is put some water molecules in there and show how water can go from an area of high water concentration or water potential to low water potential. So if you look at the concentration of water here, more on the inside, less on the outside, so water will flow from in to out. Now, if the opposite were true, water could flow into the cell, depending on the concentration of water right there. So you could represent it that way, or on your student modeling packs. This is aquaporin right here, and you can have water moving across. Now, this is good to show students in a 3D format how water moves, but it's kind of just a 30-second demo of water moving across the cell membrane. It's not as interesting as if we had some kind of scenario or example to give students. So one of the most common ones that we use in a biology classroom is talking about water movement in different, when you put cells in different solutions. So let's go ahead and take your cell membrane models now and showing you let's put sodium in our model. So I'm going to throw some of my blue sodium ions in here to demo, have students demonstrate with their models what would it look like for the amount of sodiums and waters on the outside versus inside of the cell if we put the cell in a hypertonic solution, isotonic solution, or hypotonic solution. And that would be the outside environment of the cell that we're talking about here. And so let's have aquaporin right there. And how would you model hypertonic? So in the chat, what would you be placing inside or outside in respective to each other? So let's take a look at that. So hypertonic solution, more molecules from, I think it's Shalena, more molecules on the outside. Okay. All right, so hypertonic solution, if we were to orient our waters and our sodiums, so Chris, if you could click the graphic there, yeah, you would have more sodium ions on the outside of the cell with respect to water and fewer sodiums on the inside. So you could orient more of those sodiums outside. Now, if we were to say that this membrane was impermeable to sodiums, which way would water move? You could also ask that question. Okay, and then isotonic solution, have your students or you guys model that. What would that look like? Where would you place the sodiums and the water molecules? Okay. All right, equal amounts in and out. Thank you. Okay, yeah, and so you would see that in terms of concentration of sodium ions, we're looking at solute concentration here. There's an equal concentration of sodium outside extracellularly as intracellularly, okay? And then last scenario, hypotonic. What would that look like? Move your waters or sodiums around to model that as well. Okay, thank you, Anitha. Less sodiums outside, yeah. So we have water on both sides and way more sodiums on the inside of the cell. Okay, so with the placement of these solutions, right when you put a cell into that solution, which direction will water flow? And now you can have students in their groups or at home with their individual student modeling packs to show you on camera or to talk with each other in class and show the actual movement of water molecules. So in this scenario of the hypertonic solution, let me go ahead in my overhead cam, showing you more sodium molecules outside. What you're going to see is that because there's more sodium molecules on the outside, it will have a lower water potential and a higher water potential inside. For those who are teaching AP biology, will want to use those terms, right? Water will move from an area of high water potential to where it has a lower water potential outside. So you can have students maybe do stop motion videos to demo showing water going through this channel towards the overall direction of outside. But the other thing you can do is have some water channels showing a little bit of water going inside, because remember, these water molecules can go both directions in aquaporins, but the net movement is most of, more water flowing out than in. So that's key to show students. And then for an isotonic solution, you have water moving back and forth across different aquaporins at equal rates going in or out. And so there's no net movement of water. And then of course, in a hypotonic solution, more water will move in than is going out. And so you'll have that cell fill with water and start to swell and potentially burst or lyse if it's an animal cell, right? So this would be a great model to show and for students with water movement. Now we have some other proteins that you can also model, the sodium channel, and you can see different representations here, including the middle one, which is actual 3D model that you can purchase from 3D Molecular Designs. And you can see when you turn it on the side like that, that there's this definite pore that goes in where sodium can transport across. And we also have gated potassium channels. You can have these gates that open and close. It's kind of fun as you have those brads on, or these, these little screws on there so that it can show the channels opening and closing right there. And also the transport of glucose. So you could talk about cells in their response to insulin signaling, that these glucose or GLUT transporter proteins are going to be carrier proteins that can loosely bind the glucose molecules and transport them into the cell. So those are some great examples of passive transport. Now let's take a look at active transport, and anything that has pump on it is going to be your active transport. So sodium potassium pump, calcium pump, proton pump, and these all require the use of ATP. So if we switch to our little model with looking at our pumps there, I'm going to put in my overhead cam here the sodium potassium pump right there, and you can see that it can move. So there's different conformational changes in the shape of this protein right here in response to signaling. And so this pump requires ATP, so you can actually take out your phosphate group and your ADP, and of course, through glycolysis and cellular respiration in a cell, we have the production or synthesis of ATP right there, which is used for a lot of different cell processes, including these pumps. So let's go ahead and back to the slideshow there. Great. Okay, so the sodium potassium pump is embedded in the cell membrane, and it's found in every single cell in animal cells. And this is important to maintain the electrochemical gradient from the outside to inside of the cell. And this is particularly used as we talk about examples with the signaling of neurons and transmission of neurons and action potentials, those electrical signals. So the next slide there shows the sodium potassium pump with the neurons and how they can maintain the electrochemical gradient. So this is a video of here the sodium's going in to the sodium potassium pump. ATP binds, and so that phosphate group is left as ATP is cleaved, okay, and that changes the shape of this protein. So then it faces the outside of the cell, and now these sodium ions can dissociate from this pump and move to the outside. And then we have potassium ions being attracted to the active site of this pump and finding their positions in there. And as the phosphate group detaches from this pump, it allows this conformational change again for this to face the inside of the cell. And then those potassium ions go into the cell. So with both the movement of sodium, you are moving both sodium against its concentration gradient and the potassiums against their concentration gradient, and it requires the use of ATP. So this is a really difficult concept to show with just a 2D picture on a slide somewhere. And this model is so great for students to be able to show the movement and the connection with ATP. And this is also great in terms of the synapse kit that 3D molecular designs have, where students can kind of station themselves along a long axon, for example, to show the movement of ions across. Okay, so our main example here that I wanted to show you something a little different is the CFTR protein, so the cystic fibrosis transmembrane conductance regulator, or CFTR protein. This gene is located on chromosome 7. The protein itself is fairly large, has 1480 amino acids. So on my overhead cam, I'm going to show you my printed version. So this one is not sold by 3D Molecular Designs. This year, one of the things I've been doing, during pandemic teaching is figuring out how to use a 3D printer. So I downloaded this file from the protein data bank for the CFTR protein right here, and managed to figure out how to print it on our 3D printer. And so this is in plastic. And you can see here, similar to the aquaporin, there is a lot of alpha helices running through here. So this is the portion of the protein that embeds into the cell membrane. This portion of the protein faces the interior of the cell. And so if we go back to the graphic in the slideshow with the CFTR protein, this has two identical subunits that's part of this. Down here, the part that's intracellular has two ATP binding sites, and it allows the transport of chloride ions from the inside to the outside of the cell. So I'm going to orient my chloride protein right there, CFTR protein, and some chloride ions. I'm going to throw in some of my homemade chloride ions in here. These are the ones that I made at home. So pink triangles right here. Some pink triangles on the outside, and definitely more ions on the inside of the cell. So the role of this protein is to transport chloride ions across so that you change the concentration of solutes on the exterior of the cell so that you have the movement of water molecules across the cell membrane too. And so you can see the water molecules right there. Okay. And they get transported out as chloride ions are transported with the CFTR protein. And so we can model that with our foam pieces. So you may not have a 3D printed protein yet, but you can model that with your foam piece. So this gated ion channel can represent your chloride ion channel right here. Or you could use the one that's shown in the graphic in the slideshow right there. But it is gated actually, so it can open like this, and chloride ions can be transported across with the signaling there. Now the challenge with this protein is that with any proteins that we have, it can acquire mutations in the genetic code. And so one of the mutations that can occur is leads to the genetic disorder called cystic fibrosis. And so let's take a look at this video right here to see how cystic fibrosis affects the lungs.
Cystic fibrosis, or CF, is a genetic disease that affects multiple organs, including your lungs, and limits your ability to breathe over time. Your main airway, called the trachea, branches into each of your lungs. The largest airway within each lung is called a bronchus. and each bronchus branches into smaller airways called bronchioles. Normally, airways are coated inside with a thin layer of fluid and mucus. The mucus catches dust and germs from the air you breathe in. Then, hair-like projections on the airway surface, called cilia, push the mucus, trap dust, and germs out of the lungs. In cystic fibrosis, the mucus is so thick and sticky that the cilia can't push it out of your lungs. This thick, sticky mucus can build up, trap germs like bacteria, and block your airways. Because of this, you may periodically have acute infections or exacerbations of the disease, causing symptoms such as increased coughing, production of mucus, and shortness of breath. As the disease progresses, long-term damage can occur in your lungs. This happens when germs, like bacteria, get into your lungs and cause chronic infections and inflammation. But there's a lot you can do to control the infections and slow the progression of lung damage. Talk to your CF Care team about how the different parts of your treatment plan can help keep you healthier. As your partner, they can help you find ways to fit your treatments into your daily care routine.
All right. So as you can see in that video, how CF, the mutation in the CFTR protein, can affect patients who have cystic fibrosis. So there's been over 2,000 or more identified mutations. The F508 deletion is the most common. And what that means is for those 1,480 amino acids, amino acid 508, which is phenylalanine, there is a mutation in which there's a deletion. So that phenylalanine doesn't exist anymore in the protein, which affects the trafficking of this protein CFTR to the membrane of cells. This is the most common mutation in 2/3 of the cases worldwide and about 90% of the cases in the US. So you can see it affects CF patients where their secretions become thick, mostly in the lungs, but also in other places like the GI tract and reproductive tract as well. Quite a few infants, when they're born in the hospital, you know, that little heel prick genetic testing that happens there is also testing for CF as one of the genetic conditions that infants can be born with. This disease is not just limited to people who are of European descent, but it is most common among Northern Europeans or those of Northern European ancestry, and it's more rare or less common in Africans and Asians. So let's go ahead and model that. So knowing that, what happens with patients who have various mutations of the CFTR protein? So in the first case, let's go ahead and show that picture, is there is a lack of the trafficking, the production of and trafficking of this protein to the surface of the cell membrane, or actually, let me go ahead and change that. Non-functional means there's a mutation in which it is produced, it may not have the right shape, or it may not be functional. It does make it from Golgi bodies in vesicles and trafficked to the membrane. So it's there present on the surface, but it is unable to function properly, or there's very little function of transporting those chloride ions out. So we're going to talk about how, if that is the case, where the protein is made, is found on the surface, but is not functional, there's a lot of drug therapies and treatments. One very successful one we're going to talk about in the next slide that can help patients with this particular mutation. Now, unfortunately, with this mutation, only 2 to 3% of CF patients have this condition and can respond to this particular treatment. The majority of cases in the United States that we see is the CFTR protein is not even fully made within the cell. And so this protein is probably made or not transported, or it is made or partially made and then broken down. There could be premature stop codons there. And so there is no protein that even makes it to the surface of the cell membrane. So you can see if there's no way for chloride ions to be transported out, this is a simplified version of what's going on with CF patients, and but there's also involves the sodium channels as well. But the basic idea is that without those chloride ions and ions on the outside, the water tends to stay on the inside of the cell. So the secretions on the external part of the cell with mucus becomes very thick because it's not thinned out with the water. And so you get a lot of those problems that CF patients experience. So scientists, because this is pretty widespread mutation occurring in some of our population in the US, in the next slide shows one kind of drug therapy for CF patients. This is ivacaftor. Ivacaftor is called a potentiator, and that means that for these patients who respond to this treatment, they have the production of the CFTR protein and it does get to the membrane, but this protein is not functional. And so ivacaftor, which you see in the picture on the right in red, it binds to the protein, changes its shape slightly, and now makes this protein functional. So it's quite effective for the 2 to 3% of CF patients who have this kind of mutation. So they're working on some other drugs to help with the trafficking of the proteins to the membrane, with the folding and the production. So some of these proteins, as they're translated, reach premature stop codons, and there are drugs they're making to help these ribosomes read through those stop codons and continue to translate the rest of the protein. So exciting things in the world of gene therapy and also just modulator therapies for patients who have genetic disorders. All right, so I hope you learned a few new things here, or how to use the membrane kit, both with individual students and student kits and classroom kits. I found this to be a really helpful model to use with my students, whether I'm in person or virtual. And thank you for your participation today.
Great. Thank you, Chris. That was a wonderful session. I didn't catch any questions in the chat, so if you have any questions for Chris, please go ahead and post them in there now. And we will be able to stay on for a few minutes after the session to answer any questions that you have. So, and you'll see that Chris used the phospholipid and membrane transport kit today in the membrane student modeling pack. She also snuck in a couple of our 3D printed models with our, which one did you show? With the aquaporin. And we do also have a couple of other ion channels as well.